to produce plant postbiotics. Thank you for introducing us to this idea of postbiotics.
So I would like to thank the organizer for giving us the opportunity to be here and bring you the greetings of my colleague Francesca Luzzatelli that unfortunately could not be here for this presentation. Okay, this is a brief introduction of what I'm going to present in this short time. First of all, introduce the concept of postbiotics and to see how this new class of product can be applied in the plant biostimulant sector. One major point that we have in classical experiments done with bacteria with plant growth promoting traits is that the activity of this bacteria is measured using morphological and yield parameters. These data are always not supported by experiment in which the ecophysiological parameters are analyzed, and at the same time the metabolome analysis is not supporting the real function of this biological system. So we have many papers in the literature focusing on PGPR that are working, for example, only in particular condition, for example, under stress or in combination with some bacteria. And industrial products that are based with this microorganism, they frequently fail to be used in agriculture sector. So we focus on our attention of one major point that is known in all biological systems. The metabolic state of the cells in some fact affect the phenotype, and this phenotype also affect the intra and interdomain cell-to-cell communication. This is true for all the interaction, human with microorganism, plant with microorganism, and bacteria with microorganism. Starting from this consideration that the molecules that are secreted in the medium are relevant for the communication, for the interaction between two biological systems. There has been recently a great attention to a new class of product named postbiotics. What are postbiotics? Are preparation of microorganisms that have been killed by inactivation and include the molecules that are produced from this microorganism. The advantage to use this type of system, it's well known in the human sector because allow to develop a new product that in some way take advantage of all the metabolic capacities of a microorganism. Starting from the original point that you can grow a microorganism in a special condition, just have only few traits expressed among the all traits that could be expressed from that type of biological system. Our work has been focusing on microorganisms that have been originally isolated from the phyllosphere of lettuce plant that were exposed to vegetal extract. And the original idea was to see what type of microorganism respond to, for example, vegetable biostimulant, and see how this microorganism could cooperate with biostimulant to enhance the effect of this product. One of the microorganisms that we isolated was a Panthoeagglomena strain that has several interesting traits. Among them, the ability to somewhere resist to several heavy metals, the capability to produce hormones like auxins, the capability to produce hydrophores, and the ability to solubilize phosphate. The first type of activity we did with this microorganism were classical approach. We use corn and tomato, and we treated with this strain, alone or in combination, to demonstrate that the ability to solubilize phosphate was valuable for the plant, increase root length, total root length, and above ground biomass. But there was a major problem with this microorganism. It was a Pantoea agglomerans, and we analyzed in detail the genomic DNA of this microorganism, and the affiliation to this species was never in discussion. Pantoea agglomerans is a well-known microorganism present in the phyllosphere and the rhizosphere of various environments. And the major point is that some of the strains belonging to this species are human pathogens. So you will never develop a product based on a microorganism belonging to this species. We did a lot of work to demonstrate that C1 strain was not a pathogen, was not a human pathogen, a plant pathogen, an animal pathogen. And doing this work, we did a lot of metabolomics to evaluate what type of molecules were produced by this strain, cultivating it under different culture conditions. And what we observed that, for example, changing the carbon source, you could get a profile of a secreted metabolite that is quite different. If you go to analyze this metabolite, the interesting thing is that you get, apart from, for example, auxin, other molecules that are known that could interact with auxin and in some way change the response of the plant to this hormone. So we focus our attention on the metabolome, and in particular on the exometabolome, on what could be a postbiotics based on Pantoea agglomerans. We analyze also different media, the sucrose minimal medium against a classical peptone medium. And what appeared was that the production of antimicrobial compounds and the production, for example, of hypoxanthine, xanthine could be changed varying the culture conditions. That means that you can prepare a product mixing metabolite that has been obtained under controlled fermentation condition on different media, just to have a cocktail of molecules that can in some way be applied as biostimulant. And the fermentation process is an important point of this production because the cultivation condition, the aeration condition, the stress affect very strongly the production of molecules that could be more or less present in the fermented medium. One point that I will try to focus on this is usually in a classical fermentation biotechnology approach, one focus on the production of one molecule, for example, auxin, and try to get the condition in which auxin is produced in the higher amount. When you produce postbiotics, the approach changes completely because you need a ratio of different metabolites that should be optimized to obtain a better effect. We have tested the exometabolome of Pantoea C1 grown on different culture conditions on different plant systems. This is the list of plant systems that we have used to evaluate the effect. And this is one of the examples that we have described and is related to hazelnut. This experiment was done in a nursery, and the application of the metabolite was done on 1,500 plants, and the data related to this analysis are referred to this number of plants. What we observed that if we standardize for the auxin content, we can get a better effect on the eradication of the cuttings, applying 1/3 of the standard concentration of auxin that is used in industrial nurseries. But our idea was to get more information, what they do, what these metabolites do on the plant. And we did a series of experiments on comparative experiment on Pyrrhus cuttings. And what it appeared were several interesting information. If you use auxin in a complex mixture of metabolite produced by Pantoea agglomerates, you have a reduction of the time required to get the root formation. A second important thing is that the callus formation is not observed if the hormone is produced from the microorganism and is provided together with the other exometabolite. And a third important observation that we did in this experiment is that the root architecture is completely different if you use exometabolite. So if you use Pantoea agglomerants postbiotic compared to the synthetic hormone. We try to understand what occurred to the plant at the molecular level, doing differential transcription for different genes related to the response to exogenous auxin. And what was the main conclusion from all this experiment? That the temporal expression profile of genes that are relevant for promoting adventitious root induction, root development, promotion of the root induction development, as in the case of F8, are anticipated in the timing, and the level are higher than in the control in which the synthetic Hib hormone was used. And another important observation that we did, analyzing the differential expression of auxin response 17, that repress root induction, is that the level of this protein is reduced earlier. And that is the reason why, for example, the callus formation is not observed when you use microbial metabolites. Another information that we did also always on the callus formation was the expression of KRP2 gene. And also in this case, what we observed is that this gene that is related to callus formation is expressed at a high level only when the metabolite provided by Pantoea are used as a biostimulant. So the general message that I would like to leave to you is that you can analyze the potential metabolic capability of microorganisms that are present in the phyllosphere, that are known, that are major component of the phyllosphere, that could never be studied to develop a biostimulant in the classical approach, using cells to develop classical microbial. but you could use the metabolite in designing product in which you optimize the culture condition and you combine fermentation technology with the metabolomics to evaluate what could be, in some way, the real contribute of the cocktail of molecules to the stimulatory effect. We analyzed, by comparative genomics, other strains of Pantoea agglomerants to check if this effect could be obtained with any strain belonging to this species, and the response was not at the same extent that we could obtain with the C1 strain. That means that the selection of appropriate strains still remain a critical point in the development of future bioproduct based on plant postbiotics. And I thank you for the attention. Just acknowledgement to the people of my laboratory and to all group that collaborated with us on this work. Professor from Tuscia University, as Giuseppe Colla, Maria Teresa Cardarelli, and Rosario Muleo, from other university, from the Research Center for Agriculture in Rome, CREA, and from two Spanish companies that are Olobion and Athens SL. Thanks for your attention.